Hi, everyone. Thanks for coming to SICB Plus to check out my talk. Today, I am so excited to talk to everybody about blinking. So blinking is truly an underappreciated behavior that I think a lot of us take for granted. Over the course of this talk, every person listening will blink on average once every 5 seconds, which is about 12 times a minute. But now that I mentioned that, you're probably either blinking more rapidly or you're trying your best to hold back from blinking. If you fall into that second category, stop. Blinking is critical for maintaining eye health and function. In many extant tetrapods, blinking serves multiple functions. One of these is eye wetting or maintaining a fluid film over the cornea, which is important for epithelial cell health and oxygenation. Another is cleaning the eye of debris, which can be critical to maintain visual acuity and limit damage. Blinking can also function to protect the eye from foreign objects that could scratch or puncture the cornea. And finally, blinking invertebrates can serve as a way to communicate or interact socially with conspecifics, which is a behavior that we humans have certainly used regularly and has even been discussed in some of Darwin's writings. And since the origin of this novel multifunctional behavior, blinking and its associated morphology has diversified across tetrapods. For example, in humans, eye closure is accomplished primarily through the movement of the upper eyelid. Well, in some turtles, the lower eyelid is actually raised to accomplish eye closure. And in many species of amphibians, reptiles, birds, and mammals, a third membrane, the translucent nictitating membrane, has also evolved as a way to serve these functions while maintaining some visual acuity. However, hypotheses of how and why this behavior originated in early tetrapods are non-existent. We believe that the blinking behavior has only evolved once in tetrapods, and that the evolution of terrestriality in vertebrates is a strong selective pressure driving the evolution of this behavior, and we've found several lines of evidence to support this hypothesis. First, to our knowledge, most fish do not exhibit a blinking behavior, although blinking has evolved in some shark species and is believed to protect the eye during predation. A second line of evidence is that in species of reptiles and amphibians that have secondarily evolved to be fully aquatic, like the species of frog on the right, blinking has been repeatedly lost in these lineages. A third piece of evidence is that in the mudskippers, there are a second line of finches, this time the actinopterygians, that have evolved to spend the majority of their day on land. And similarly, mudskippers have convergently and independently evolved blinking as a behavior. So at this point, we know mudskipper blinking is phenomenologically analogous to blinking in tetrapods, but is this behavior actually convergent? To support a hypothesis of convergence, we need to know how and why blinking has evolved in mudskippers. And for the remainder of this talk, we're going to be doing just that. First, we're going to focus on the kinematics and movement of the eye, and then take a deeper dive into the associated morphology to see what morphological adaptations have evolved with this behavior. Next, we'll focus on determining the function of blinking in mudskippers. And based on what we know from blinking in humans, we're specifically going to test the hypotheses in mudskippers that blinking functions to wet the eye in the corneal surface, to clean the eye from foreign debris, and finally, protect the eye from puncture by foreign objects. And before we continue, I want to recognize the amazing group of undergraduates that have conducted much of the work you're about to see through their experience in a multi-semester vertically integrated project course at Georgia Tech, where authentic research experiences are integrated directly into the curriculum. And this is a really innovative educational activity that reduces barriers for undergraduates seeking research experience, especially those that belong to underrepresented minorities in STEM, and also those of a lower socioeconomic status. And over the last 3.5 years, throughout this course, either this project or others in the course, over a quarter of the students that have participated in this class will actually have published products. And I think that is incredibly awesome and cool. So let's dive into the how. The first thing we did was examine the kinematics of mudskipper blinking. We can see in the blue trace of this figure, mudskipper blinking is achieved by the depression or retraction of the eye itself into a ventrally located pocket. And as the eye moves downward, a membrane below the eye, which is known as the dermal cup, and highlighted in magenta in this figure, is displaced laterally and dorsally to help cover the entirety of the eye globe. And to investigate how this behavior is accomplished and whether any new musculature, glands, or secretory cells have evolved in tandem, we conducted contrast-enhanced CT scanning of the soft tissues of the eye apparatus. And we find that mudskippers have not evolved any new musculature to accomplish this behavior and rely on some combination of their 6 extraocular eye muscles to actuate the eye. And these are the same 6 muscles that are well conserved across vertebrates. Next, we further analyzed the scan and determined that mudskippers have not evolved any tear glands that could be analogous to the lacrimal or meibomian glands of humans and other vertebrates. And we also confirmed this through histological staining and imaging of several areas around the eye and other parts of the body, and again found no evidence of glands or a high concentration of secretory cells. We do find the presence of the mucus-secreting goblet cell, which are those highlighted in the bottom right-hand corner of the histological section, indicated by red arrows, but they aren't in any higher concentration here in the dermal cup than they are in other aspects of the body. So we find that in mudskippers, blinking is achieved through the ventral retraction of the eye into the orbit, where it's passively covered by a ventrally located membrane. And this demonstrates that a complex behavior can evolve in systems with rudimentary morphology and suggests that few morphological changes or specializations are needed to occur for this behavior to evolve. So while we've gained insight into the mechanics of blinking and the associated morphology that facilitates this behavior in mudskippers, we don't yet know the functional roles blinking plays in mudskippers and whether it serves the same purpose as it does in other tetrapod systems. So the next question we wanted to ask was, does blinking function to wet the eye? To do this, our team created an experimental chamber that was equipped with a computer fan at the top and a small pool of water at the bottom of the tank. Mudskippers were left in this tank for a total of 2 hours. For one of the hours, the fans were off, and for the other hour, the fans were turned on. We calculated that turning the fans on increased the evaporation rate of a thin fluid film by approximately 20 times. To ensure the length of the experiment did not impact our results, we alternated whether we turned the fans on for the first or the second half of the experiment. And when we reviewed the film, we identified the timestamp of each blink throughout the test period. We calculated the interblink interval, the IBI, which is the duration of time between 2 consecutive blinks. And we then compared that between our 2 conditions, fans on versus fans off. And we would predict that the increase in evaporation rate within the chamber should increase blink rate and therefore decrease the interblink interval. And that's exactly what we see. We find that the interblink interval is significantly shorter in the high evaporation conditions. So mudskipper blink rate goes from about 1 blink every 2 minutes in control conditions to 2 blinks in a single minute when under high evaporation. So together, these data support the hypothesis that one function of blinking in mudskippers is to wet the eye. Next, we wanted to test whether another function of blinking is to clean the eye. This is important because mudskippers, as you might have guessed from the name, they love the mud, and keeping the eye clean is critical for maintaining visual acuity, for locating predators, prey, and conspecifics. To test this hypothesis, we conducted a very simple experiment. We sprinkled as many brine shrimp eggs as we could over the corneal surface. We chose brine shrimp eggs because they're approximately 200 microns in diameter, which is within the size range of sand and dirt particles found in waterfront environments. And this is a completely harmless foreign particle to the mudskippers. And once we sprinkled these eggs over the eye, we just waited for the mudskipper to blink. And then we reviewed videos of these experiments to calculate the number of egg particles covering the corneal surface before and after a blink. And when we consider all the blinks we recorded, we find that a single blink is highly effective at cleaning the corneal surface, and in most cases could completely clean the eye surface of all of its debris. So we can now also support our second hypothesis that another function of blinking in mudskippers is to clean the eye. The third functional hypothesis that we wanted to test was that blinking can also serve to protect the eye from puncture. To do this, we created a capacitance sensor, like one within the screen of your phone. And when the sensor touches a conductive surface, like the body of an animal, it would illuminate a small LED, which we also kept in view. And we filmed ourselves using a high-speed camera lightly tapping the upper corneal surface of the eye, which would reliably trigger a blink. But we wanted to take this a step further to determine if this behavior could be a reflex. So from these videos, we calculated two variables. The first was the lag time between when the eye was tapped and when the blink was initiated. And next was the duration or speed of the eye closing period, which is the duration it takes the eye to become fully depressed during the blink cycle. And these metrics have been measured for the corneal reflex in humans, which is the only other system we found data for this reflex. So we could compare these data to determine if the temporal patterns of mechanically stimulated blinks in mudskippers are similar to reflexive blinks in humans. And what we find is that the lag time averages just under 30 milliseconds in mudskippers. And this is very similar to the lag time of the corneal reflex in humans. We see a similar average, and we see a similar degree of variability across these data. Next, we found that the duration of the depression phase, or the duration it takes the eye to close, is significantly shorter and therefore faster in mechanically induced blinks in comparison to spontaneous blinks, which we had multiple samples of from our previous kinematic work. And in humans, we see similar trends. The duration of the down phase of the upper eyelid is shorter in reflexive blinks in comparison to spontaneous. And the velocity is also greater, which you could see by comparing the data on the left versus the right half of these four panels for the human blinking. So combined with the high likelihood that fully aquatic relatives of the mudskipper have not evolved any sort of eye retraction given their anatomy, we can now suggest this represents the evolution of a novel reflex. So we can now also support our third hypothesis that another function of blinking in mudskippers is to protect the eye, which has likely occurred through the evolution of a novel reflex loop. So by studying this extraordinary behavior in a system where it is convergently and independently evolved, I think we can start to make some hypotheses of different ways this behavior could have originated in tetrapods. The first thing that we demonstrated is that a complex behavior can evolve in systems with rudimentary morphology. This suggests that few morphological changes or specializations are needed to occur for this behavior to evolve in early tetrapods, and that the origination of this behavior could have occurred through a simple eye retraction mechanism similar to what we see in mudskippers. And this is because most reconstructions find that the orbits of early tetrapods are actually on the dorsal aspect of the skull. Next, it demonstrates that it's possible for multiple functions to evolve through the evolution of a single behavior. In mudskippers, we saw three totally distinct functions are being performed with great efficiency by deploying the same behavior and by using the same associated morphology and neural control across all three functions. This suggests that it's possible for multiple functions to evolve as a suite rather than as a stepwise process, and that multiple independent selective pressures for different functions can simultaneously drive the evolution of a single multifunctional behavior. And because of that, that's why this relatively complex behavior can evolve independently and repeatedly in lineages separated by many millions of years. And with that, I'd like to thank everybody in the Sponberg lab, the Shubin lab, the Baumla lab, the Georgia Tech Animal Care team, and so many other colleagues that have helped along the way. And I want to thank everybody that came to the meeting, that is hanging out here on Virtual Plus, and I especially want to thank the organizers of SICB, Melina, Jake, Brett Burke, and so many other people that have helped along the way. This has been incredibly awesome, and I can't wait to continue experiencing this meeting here on SICB Plus. Thanks for checking it out. Please reach out to me here on Twitter or email if you have any questions or comments about the talk. Take care, everybody, and I'm looking forward to seeing everybody next year. Bye.